SOS response. Despite having multiple repair systems, sometimes the damage to an organism's DNA is so great that the normal repair mechanisms just described. As a result, DNA synthesis stops completely. In such situations, a global control network called the SOS response is activated. The prokaryotic SOS system is regulated by two main proteins, LEX-A and REC-A. The most common cellular signals activating the SOS response are regions of single-stranded DNA, ssDNA, arising from stalled replication fork or double-strand breaks, which are processed by DNA helicase to separate the two DNA strands. During normal growth, the SOS genes are negatively regulated by LEX-A repressor protein dimers. For initiation of SOS response, REC-A protein binds to ssDNAs in an ATP hydrolysis driven reaction, creating REC-A to ssDNA filaments. REC-A binds to single or double-stranded DNA breaks and gaps generated by cessation of DNA synthesis. REC-A binding initiate recombination repair. REC-A and ssDNA filaments activate LEX-A. REC-A ssDNA filaments activate LEX-A autoprotease activity, which ultimately leads to cleavage of LEX-A dimer and subsequent LEX-A degradation. The loss of LEX-A repressor induces transcription of the SOS genes and allows for further signal induction, inhibition of cell division, and an increase in levels of proteins responsible for damage processing. The first SOS repair mechanism to be induced is nucleotide explosion repair, whose aim is to fix DNA damage without commitment to a full-fledged SOS response. SOS repair system operates only under potentially lethal conditions caused by extensive UV radiation. Therefore, SOS repair system works on the principle that survival with mutation is better than no survival at all. This repair is also called error-prone repair. So, REC-A of LEX-A, low-level expression, target genes, DNA damage inducing signal, REC-A protein activation, LEX-A repressor cleaved, LEX, then signal activated REC-A, DNA repair and other SOS functions, 20 target genes. Next topic, antisense RNA strategies for regulating gene expression. Antisense RNA, AsRNA, also referred to as antisense transcript, natural antisense transcript, or antisense oligonucleotide B, is a single-stranded RNA that is complementary to protein coding messenger RNA or mRNA, with which it hybridizes and thereby blocks its translation into protein. The AsRNAs, which occur naturally, have been found in both prokaryotes and eukaryotes and can be classified into short nucleotides and long, greater than 200 nucleotides, non-coding RNAs or ncRNA. The primary function of AsRNA is regulating gene expression. AsRNA may also be produced synthetically and have found widespread use as research tools for gene knockdown. They may also have therapeutic applications. Antisense RNA molecule represent a unique type of DNA transcript that comprises 19 to 23 nucleotides and is complementary to mRNA. Antisense RNA play a crucial role in regulating gene expression at multiple levels, such as replication, transcription, and translation. Antisense RNA technology is one of the approaches that are used for the inhibition of gene expression or downregulation of a gene. This technology works on the principle that an antisense nucleic acid sequence base pairs with its complementary base RNA strand and prevents it from being translated into protein. The complementary nucleic acid sequence can be either a synthetic oligonucleotide, often oligodeoxyribonucleotides or less than 30 nucleotides, or longer antisense RNA sequences. The complementary sense and antisense RNA molecule in the same cell can lead to the formation of a stable duplex, which may interfere with gene expression at the level of transcription, RNA processing, or possibly translation. Antisense refers to the orientation of complementary strands during transcription. The two complementary strands of DNA are referred to as sense, coding or plus, and antisense, non-coding or minus. Transcription uses the antisense strand as template, resulting in an mRNA that is identical in sequence to the sense strand, except for the replacement of uracil or thymine. Antisense RNA is synthesized using the sense strand as template, therefore it has a sequence complementary to mRNA. So sense strand, antisense, transcription sense. So later, normal mRNA is formed, antisense mRNA is formed. Normal and antisense RNA can anneal. So antisense RNA sequences are complementary to a target mRNA. Antisense RNA forms double-stranded regions that block either protein translation or slicing of introns. Antisense RNA technology is a method used to silence genes and inhibit their expression. Gene function relies on expression, which follows the central dogma of molecular biology. The use of AsRNA, antisense RNA, can regulate gene expression by targeting specific mRNAs, which can result in changes in phenotype, disease resistance, and other traits associated with protein expression profiles. This technology uses short, single-stranded oligonucleotide strands that are complementary to the target mRNA. Antisense technology works on the principle of complementary base pairing between antisense oligonucleotide sequences and its corresponding sense RNA strand. This binding impedes translation, the ultimate step in protein synthesis, by blocking the sense RNA strand from participating in the process. The messenger RNA or mRNA is responsible for transmitting genetic information from the DNA to other cellular components involved in protein production. To put it simply, antisense technology uses complementary base pairing to halt protein production by interfering with mRNA function. The antisense approach aims to decrease the expression of a specific molecular target. This is accomplished by triggering the activity of RNase H endonuclease, which cleaves the RNA duplex formed between the antisense RNA and the mRNA of the target gene in the cytoplasm. This cleavage leads to a reduction in protein synthesis and a significant decrease in the translation of the target gene. The primary strategy for ASONs or gapmers to trigger RNase H activation, which cleaves the mRNase RNA portion and antisense RNA duplex, resulting in the degradation of the target mRNA. Furthermore, ASONs that do not cause RNase H cleavage can impede translation by physically obstructing the ribosome, known as steric blockers. When targeting the 5' terminus with ASONs, it is possible to prevent the binding and assembly of the translation machinery. Then antisense RNA regulation strategies. Antisense RNA strategies are a set of techniques used to regulate gene expression by targeting specific RNA molecules. Antisense RNA strategies are a set of techniques used to regulate gene expression by targeting specific RNA molecules. These strategies involve the use of antisense oligonucleotides or antisense RNA molecules to interfere with the expression of target genes. Here are a few commonly used antisense RNA strategies for regulating gene expression. Antisense oligonucleotides or ASOs. ASOs are short, single-stranded DNA or RNA molecules that are designated to be complementary to a specific target DNA sequence. By binding to the target RNAs, ASOs can prevent translation or promote degradation of the RNA molecule, thereby reducing the expression of the corresponding gene. ASOs can be delivered into cells using various methods such as transfection or viral vectors. Small interfering RNAs, siRNAs. siRNAs are double-stranded RNA molecules typically generated from longer hairpin precursor molecules. Once inside the cell, siRNAs are processed into small RNA duplexes that guide the RNA-induced silencing complex to the target mRNA. The RISC complex then cleaves the target mRNA or inhibits its translation, leading to reduced gene expression. Then short hairpin RNAs, shRNA, are designed to mimic the structure of naturally occurring microRNAs. They are usually expressed from DNA plasmids or viral vectors and processed in the cell to generate small hairpin RNA molecules. These hairpins are then cleaved into small interfering RNAs that mediate gene silencing through the RISC complex, similar to siRNAs. Then antisense mediated exon skipping. This strategy is commonly used to modulate splicing of pre-mRNA molecule by targeting specific splice sites of exotic sequences. Antisense oligonucleotides can disrupt the normal splicing process, leading to the skipping of specific exons during mRNA maturation. Exon skipping can be used to correct genetic mutations or modulate gene expression by removing specific exons from the mature mRNA. Then antisense RNA based gene activation. In addition to gene expression, antisense RNA molecules can also be used to activate gene expression by designing antisense molecules that target non-coding regulatory elements or promoter regions. It is possible to modulate gene transcription and enhance gene expression. These antisense RNA strategies offer powerful tools for studying gene function, modulating gene expression levels, and developing potential therapeutic approaches for various genetic disorders. However, it is important to consider factors such as delivery methods, stability, and specificity when designing and implementing antisense RNA-based strategies. siRNA and miRNA in regulation. The down regulation of gene expression by miRNA and siRNA is a complex process involved both translation repression and accelerated mRNA turnover, each of which appears to occur by multiple mechanisms. Moreover, under certain conditions, miRNA is also capable of activating translation. A variety of cellular proteins have been implicated in these regulatory mechanisms, yet their exact roles remain largely unresolved. Small interfering RNA, sometimes known as short interfering RNA or silencing RNA, is a class of double-stranded RNA at first, non-coding RNA molecule, typically 20 to 24, normally 21 base pairs in length, similar to miRNA, and operating within the RNA interference pathway. It interferes with the expression of specific genes with complementary nucleotide sequence by degrading mRNA after transcription, preventing translation. Schematic representation of siRNA molecule, 19 to 21 base pair RNA, core duplex that is followed by a 2 nucleotide 3' overhang on each strand, 3' hydroxyl 5' phosphate. Naturally occurring siRNAs have a well-defined structure, that is a short double-stranded RNA with phosphorylated 5' ends and hydroxylate with 3' ends with 2 overhanging nucleotides. Ricer enzyme catalyzes production of siRNAs from long dsRNAs and small hairpin RNAs. siRNAs can also be introduced into cells by transfection, since in principle any gene can be knocked out by a synthetic siRNA with a complementary sequence. siRNA are an important tool for validating gene function and drug targeting in the post-genomic era. The siRNA silence genes by inducing degradation of the target mRNA, while the miRNA tend to silence genes by interfering with accumulation of the protein products of the target mRNA. However, if base pairing between an animal miRNA and its target mRNA is perfect or near perfect, the miRNA can cause cleavage of the target mRNA. The siRNAs are formed by dicer action on double-stranded RNA that usually contain at least one strand, that is foreign to the cell or derived from transposons. On the other hand, the miRNA are formed by dicer action or the double-stranded part of a stem loop RNA, that is a normal cellular product. The siRNA base pair perfectly with the target mRNA, whereas the miRNA usually base pair imperfectly with their target mRNA. Then mechanism of siRNA action. The method by which siRNA causes the silencing of genes is as follows. Double-stranded RNA is cleaved by the diacer enzyme. This follows siRNA. The double-stranded siRNA enters the cells and forms the RNA-induced silencing complex with other proteins, that is unwound, which forms a single-stranded siRNA. The strand of RNA with the 5' end base pairing, that is thermodynamically less stable, remains part of the RISC complex. This strand can now scan for complementary mRNA. Once this antisense strand binds to the target mRNA cleavage is induced. The foreign mRNA is recognized by the host cell as abnormal and is degraded. No translation is possible and therefore the gene is silenced. siRNAs have very tight target specificity as they cleave the mRNA before translation compared to the similar miRNA, which silences genes by repression translation. siRNA has 100% complementarity or its target mRNA. siRNA has also been found to activate gene expression. The mechanism by which natural siRNA causes gene silencing through repression of translation occurs as follows. Long dsRNA, which can come into hairpin complementary RNAs and RNA-dependent RNA polymerase, is cleaved by an endoribonuclease called dicer. Dicer cuts the long dsRNA to form short interfering RNA or siRNA. That is what enables the molecule to form the RNA-induced silencing complex. Once siRNA enters the cell, it gets incorporated into other proteins to form the RISC. Once the siRNA is part of the RISC complex, the siRNA is unwound to form single-stranded siRNA. The strand that is thermodynamically less stable due to its base pairing at the 5' end is chosen to remain part of the RISC complex. The single-stranded siRNA, which is part of the RISC complex, now can scan and find a complementary mRNA. Once the single-stranded siRNA part of RISC complex binds to its target mRNA, it induces mRNA cleavage. The mRNA is now cut and recognized as abnormal by the cell. This causes degradation of the mRNA and in turn no translation of the mRNA into amino acid and then proteins, thus silencing the gene that encodes that mRNA. siRNA is also similar to miRNA. However, miRNA are derived from shorter stem loop RNA products. miRNA typically silence genes by repression of translation and have broader specificity of action, while siRNAs typically work with higher specificity by cleaving the mRNA before translation with 100% complementarity. So dsRNA, hairpin, siRNA duplex, dicer, formation of RISC, AGO binding, siRNA mRNA complex, silenced mRNA, gene silencing. Then miRNA, another class of small RNA called microRNAs are the 22nd tRNAs produced naturally in plant and animal cells by cleavage from a larger stem loop precursor. In animals, these miRNAs then base pair through, though imperfectly, with the 39 untranslated regions of specific mRNA and silence gene expression primarily by blocking translation of those mRNAs. miRNA are short non-coding RNAs that regulate gene expression post-transcriptionally. They generally bind to the 3' UTR untranslated region of their target mRNA and repress protein production by destabilizing the mRNA and translational silencing. MicroRNA is the name of a family of molecules that helps cells control the kinds and amounts of protein they make. Thus, cells use microRNA to help control gene expression. MicroRNA or miRNA, a subset of non-coding RNA, are 22 nt long endogenously initiated short RNA molecules that are considered to post-transcriptionally regulate the cleavage of target mRNAs or just to repress their translation. miRNAs are thought to be important regulators of gene expression and act via dynamic mechanisms that are influenced by molecular localization and mechanisms of interaction with targets. Then mechanism of action of microRNAs. miRNA binds to target mRNA through sequence complementarity, leading to changes in the translation pattern of mRNA proteins. miRNA bind to their target primarily at untranslated region 3' UTR. Alternately, miRNA bind to other regions such as 5' UTR or even to promoter regions in the target mRNA. Binding site determines the biological consequences of these interactions. Most miRNAs inhibit gene expression by hindering target mRNA protein translation. Interactions between miRNA and target mRNA may lead to mRNA degradation through cleavage mediated by an endonuclease in RISC to which the mRNA is bound. In animal cells, most miRNA are only partially complementary to target mRNA, which seems to be enough to permanently inhibit target mRNA translation. The small size of miRNA and the tolerability of partial complementarity with its targets allows interaction with many mRNAs. The major difference between siRNA and mRNA is that the former inhibit the expression of one specific target mRNA, while the latter regulate the expression of multiple mRNA. siRNA and mRNA inhibit translation by two different mechanisms while working in association with the protein, forming a ribonucleoprotein complex called RNA-induced silencing complex or RISC. The proteins in RISC unbind siRNA and remain bound to a single antisense strand, which then binds to mRNA in a sequence-specific manner, in which time a protein component of RISC called a silencer cuts the mRNA in the middle of the binding region. The cut mRNA is recognized by the cell as being abnormal and is subsequently destroyed. In the case of miRNA, a microRNA induced silencing complex associated with the mature mRNA, and the complex binds to mRNA and physically blocks translation. Many miRNAs form imperfectly complementary stem loop structures on the target sense strand of mRNA, as opposed to siRNAs, which require near perfect matches. In general, only one mRNA is produced from one precursor. In contrast, siRNA is proposed to moderate its own amplification in plants and certain animal species, such as C. elegans. Proposed model suggests that either the single-stranded mRNA siRNA hybrid or the sense strand of siRNA, which is released by RISC, undergo elongation or transcription respectively by RNA-dependent RNA polymerase to generate a new double-stranded piece of RNA, which acts as a new substrate for dicer and can ultimately lead to formation of new RISC.